All right, everyone. So we are going to go ahead and get started. And first off, wherever you are calling in from, let me say good afternoon, good evening, good morning, welcome. We are truly overjoyed to have you here on this Giving Tuesday. We are very honored. We feel very blessed just to have you in attendance to be part of this webinar for the next hour or so. And for those of you who I have not had the pleasure to meet or speak with yet. My name is Clark Rockway. I am the Senior Manager of Fundraising and Growth Strategy at MPN Research Foundation. Today, I will be your host as we look at our past, the present, and progress into the future as we discuss shaping the future of MPN research. But before we get started, I did want to give you a brief overview of what you can expect to hear about today. First, we will briefly dive into the past to learn about the history of how MPN Research Foundation came to be, while also remembering those who have helped pave the way. We're also going to highlight some of our ongoing research as well as our current impact. And from there, we'll have our keynote speaker, Dr. Marina Kremenskaya, a world-renowned MPN researcher, about some of her ongoing and future research activities and what has her excited. Additionally, as we celebrate the past 25 years at MPN Research Foundation, we look forward to the future and what progress looks like as we head into 2025 and beyond. And then finally, I would like to highlight that today is Giving Tuesday, and with that, our board of directors have generously met our $75,000 matching challenge. So every dollar given today will be matched up to $75,000, allowing you to double your impact with your gift. And then additionally, before we dive in, I do want to be sure to mention that we are happy to help guide and provide resources and education, but please know that we cannot provide medical advice or answer any questions related to direct care or your diagnosis. These questions should be addressed with your medical providers. We do, however, encourage you to place your questions in the Q&A feature of the webinar. A member of our team will be answering a member of our team will be answering these questions as I present. The chat feature should only be used to communicate with one another. If it is a question, please put it in the Q&A feature of the Zoom webinar, and any questions that we don't have a chance to get to today, we will follow up with an email. So to get into the mission history and some of the research activities, we do have to go back to the beginning. And this stems all the way back to 1997 when Robert Rosen, who's pictured here on the right, was diagnosed with polycythemia vera in 1997. He pulled together the few MPN patients that he could find and established the MPN Research Foundation in 1999. And you may already be familiar with our mission, but if not, our mission at the MPN Research Foundation is to stimulate original research in pursuit of new treatments and eventually a cure for the blood cancers polycythemia vera, PV, essential thrombocythemia, ET, and myelofibrosis, MF, collectively known as myeloproliferative neoplasms, or MPNs. We do this through a combination of investing in innovative research, advocating for the patient voice, and convening the global MPN community by bringing together patients, researchers, and industry around the common goal of realizing new treatment options and ultimately cures for MPNs. We are committed to funding research in their quest to treat and ultimately cure MPNs, and we're also committed to providing education and resources to patients, their families, and the entire MPN community. Working together, we really can change the prognosis and create a brighter future for people whose lives are affected by this group of diseases. And as I just alluded to, at MPNRF, we are both patient and research centered. We actually have three unique roles in the MPN community that help us to advance the research mission. First is the research itself. We identify, fund, and support novel research to accelerate the discovery and development of effective MPN treatments. Secondly, we convene, and what I mean by that is that MPNRF helps to facilitate global engagement across patient and caregiving, research and clinical and industry stakeholders. And then lastly, we advocate. We help to advance patient perspectives and address researcher challenges in discovery development. But of course, we are unable to do any of this alone. We have a wonderful community dedicated to helping one another and to advocate, which is huge because we can't do this alone. And speaking of advocacy, I would like to take this time to introduce one of our board members, Pam Murphy, to speak about Zhenya Senyak, who not only made an impact on her life, but on the life of so many others as well. So welcome, Pam. The floor is yours.
Greetings, and I'm so happy to be here to speak about Zhenya. He was one of the original advocates for MPN, good friends with Bob Rosen and our chairman of the board, Barbara Van Heusen. He was probably the most tenacious, most passionate, most opinionated, and most demanding advocate I ever met. And I worked with a lot of patients as the communication director at Insight at the time when Insight had approval of ruxolitinib or Jakafi. And Zhenya was remarkable in his tenacity to try to cover the story for the MPN community. And when we had received FDA approval, all he wanted from me was a picture of the Jakafi bottle, which I think you can see on your screen, hopefully, at the corner. We weren't approved. We were, and we were just approved. We weren't allowed to do any of that promotional stuff without the product label fully available. So his comment to me was, when I refused to do that, "You are the worst PR person I have ever met in my life." That said, he did more for the MPN community at the early time of its foundation, of its beginnings, and we hope we make you proud, Zhenya, in the future of MPNs. Thanks, Clark.
Thank you so much, Pam. Really appreciate that. Now, moving forward, I do want to talk about our unique position at the intersection among three key stakeholders: patient caregivers, researchers and clinicians, and the biotech and pharma industry. This all is part of the MPN Research Foundation research strategy as a whole. We facilitate a collaborative approach to research that helps to elevate the patient voice in research and drug development. It brings disease experts together, and it helps to accelerate the industry's understanding of an extremely complex group of diseases. And here at MPNRF, we have a proven track record of investing in meaningful projects. Our portfolio approach to evaluating high-risk, high-reward research projects leverages experts in a robust peer review process that mirrors the NIH. And over the last 25 years, we have invested over $20 million in research across 105 research projects. Throughout that funding, 99 researchers were supported in 9 countries at 49 institutions. Our patient caregiver community is constantly growing, and today we are connected to over 14,000 patient caregivers. And in that time, MPN Research Foundation has been a part of some incredibly exciting projects and advancements. Some of this includes being the first to locate and identify genes that lead to the MPNs, so driver mutations like the JAK2, the MPL, and CALR. The identification of preclinical drug candidates that selectively eliminate JAK2 mutated cells. Groundbreaking research showing MPN driver mutations that were often acquired decades before diagnosis. The identification of the CALR mutation as MPN disease driver and support for the first CALR vaccine-based MPN clinical trial in the US. A significant commitment to disease progression and established progression research network, and advancing understanding of interferon and individual patient responses. Now, of course, this is not all encompassing, but it does show how we are continuing to advance and progress every day towards our goal of finding a cure for MPNs. And we're able to have such an impact because of so many dedicated staff and volunteers. Here's a snapshot of our team, a diverse group of expert and specialists in their field. And together we form a small but mighty team dedicated to building on a 25-year legacy of impactful research investment. Now, we also have our Patient Impact Council, which is composed of patients and caregivers spanning a diverse range of MPN experiences, diagnoses, demographics, and geographic locations. This council supports MPNRF's commitment to patient-centric MPN research. And then finally, we also have our Scientific Advisory Board that MPN Research Foundation actively engages with. This Scientific Advisory Board strategically helps us to develop a research agenda and effective funding mechanisms. But in addition to the advisory boards and impact councils, we are lucky enough to work with some other incredible researchers and experts in the MPN field. One of those incredible researchers is none other than Dr. Marina Kremenskaya, who we are lucky enough to have on the call with us today and is our keynote speaker. Now, Dr. Kremenskaya is a world-renowned MPN researcher, a leading expert in MPNs focused on innovative research and treatment advances, passion for patient-centered progress. She's committed to uncovering new therapies and improving patient care, advancing future breakthroughs, and working on cutting-edge research with a vision to transform the future of MPN patients. So welcome, Dr. Kremenskaya.
Hello, everyone, and thank you so much for the invitation to speak here today. It's an honor, and thank you for the kind words. So I'm gonna speak today about the project that I've been working on at Mount Sinai, together with a group of many talented scientists and clinicians to move this forward. Next slide, please. So I have to give a little background first, even though I'm sure many people on this call are aware of this information. But as you're aware, MPNs are a group of heterogeneous disorders. And so on this one end of the spectrum, we have polycythemia vera, essential thrombocythemia, and prefibrotic myelofibrosis. And these disorders are characterized more by vascular events, such as thrombotic events, strokes, and heart attacks, and other blood clots, as well as symptoms that can often be debilitating to patients, such as fatigue and itching, what we call pruritus, difficulties concentrating, and many others. And the next step in the spectrum is myelofibrosis, as well as post-essential thrombocythemia and post-PV myelofibrosis. And here we see progressive constitutional symptoms such as, again, fatigue, bone pain, night sweats, weight loss, and many other symptoms, as well as increase in spleen. And here we often worry about low blood counts. And then at the very end of the spectrum, we have leukemic transformation, where life expectancy can be very short. And now, unfortunately, patients can progress to leukemia from any of these early diseases. That usually happens from myelofibrosis to leukemia transformation. However, this is a main point of research where we really want to figure out how to stop this progression that we see here and to never see leukemic transformations in our patients. Next slide. So what we know about these disorders is that we have what we call driver mutations. So majority of patients will have either JAK2, MPL, or calreticulin mutations, and we know that patients with polycythemia vera, most of them will have the JAK2 mutation, and for both ET and myelofibrosis, we have a combination of everything. But this is where we really start seeing CALR as can be seen in a big proportion of patients with ET as well as with myelofibrosis. Next slide, please. And so the major pathophysiology of MPN, so the major driving force behind the MPN is these driver mutations. Next, please. And what we see is that this increased JAK-STAT signaling, and it's happening in a constitutive manner. And what that means is that it's always on. So normally in the normal process, these molecules that you see drawn here on the surface of the cells, when they are engaged, these receptors, when they're engaged by signaling molecules such as a hormone, for example, erythropoietin or other hormones, that signal gets transmitted to the cell and eventually to the nucleus and to the DNA. And that signal is what should result in activation of the cells. However, what we see in MPN is that activation of this JAK-STAT signaling pathway is happening whether the outside signal is coming in or not. And that can be through the, like, for example, through the JAK2 mutation, from the calreticulin mutation, through mutation in the MPL, which is a thrombopoietin receptor. And I will talk specifically a little bit more about calreticulin in a second. Next slide, please. So calreticulin is one of the driver mutation. It's something that was discovered later than the others. And what was realized is that a number of mutations can happen in calreticulin protein, but they all fall into this C-terminal domain of the protein. And what happens is that it enables calreticulin molecule to bind to the MPL receptor and bring it to the surface of the cell and allow it to signal constitutively, again, meaning just always on, and that results in this, what we call oncogenic transformations, where the cells are undergoing division and survival when perhaps they shouldn't be. Next slide. And this is a very busy slide that I took from one of the review papers, but what I wanted to show here is that there are many different targets that are being studied currently, both in lab as well as in clinical trial, to try to get to the underlying pathophysiology of MPNs. And so what we're trying to do is get to the mutated stem cells and prevent them from undergoing self-renewal and division, and allow normal stem cells to proliferate and allow normal blood cell production to take place. And so these are being targeted with different molecules that are targeting the inflammatory milieu, for example, or the fibrosis that happens in the bone marrow. That's the A picture. The B picture is where we're targeting all the different signaling molecules that we know are important in pathogenesis of MPNs. For example, the different JAK inhibitors, of course, are targeting directly to the JAK2. And then there are multiple targets that are targeting the apoptotic pathway. So this is the pathway that tells the cell to stop surviving and you should undergo cell death. And it's a very important cellular process where in many malignancies, this process doesn't happen as it should. And then, of course, the telomerase inhibitors, epigenetic regulators. So these are molecules that are important for appropriate DNA function and transcription of the DNA into proteins. So very, very exciting work happening right now, like I said, both in lab and already in clinical trial targeting these molecules. Now in picture C, we're looking specifically at the CALR and where the mutated CALR is being targeted by the antibody that is going to interrupt this interaction between the CALR and the MPL receptor. And hopefully that would mean that the ongoing signaling is going to be interrupted. And then in D is the vaccine approach, where we're going to teach our immune system to go after mutated MPN cells and keep them under control. And this is the approach that I'm going to be talking about more. But what I wanted to point out is that there are many, many different approaches to target MPNs and the abnormalities that happens in MPNs. And what we're seeing now is that these different approaches also are being studied in combinations. This is what we're going to see more and more in the future. And I think as this field develops more, as we see with other cancer therapies is that we will be using combination approaches to really control the disease and stop it from progressing. Next slide, please. So a little bit again about the CALR specifically is that many different mutations can happen in MPNs, specifically in this molecule's CALR. But regardless of the mutation type, it results in this unique epitope that is shared in all patients with mutated CALR. And what that means is that there is a part of the protein that exists now only in patients who have these CALR mutations. It does not exist with normal CALR. And then what we call that is this results in MPN-specific shared tumor neoantigen that could potentially elicit anti-tumor immune response. And so again, what happens is that there is a protein generated that does not exist in normal cells, and it's expressed on the cell surface. Next slide, please. So immunotherapy is something that have been a very active field of investigation for oncology in general, and there are currently many immunotherapy options for cancer in different types of diseases and that are very effective and are being used to a large extent, and I think everybody knows that. We don't have a lot of immunotherapy approaches for MPNs right now. And the goal of many researchers is to see how we can use the immune system to really get to the, again, to the underlying pathology of MPN, where we want to get to that abnormal stem cells. However, we have been using immunotherapy in MPNs to some degree for a long time. For example, stem cell transplant, of course, is the only curative option right now that we have right now for MPNs. And we have been using that for many decades and successfully to cure MPNs. We have been using interferon and are using interferon successfully to treat MPNs. But the other approaches are shown here is, for example, immune checkpoint blockade, as well as antibodies and the vaccine therapies to try to get to the pathology of MPNs. Next slide. So, seeing that mutated CALR results in this novel protein that is expressed on the surface of cells in MPNs, a group of very smart scientists got together, and this included scientists who were working on cancer immunotherapy, as well as scientists and clinicians who specialize in MPN, saying this could be potentially a great candidate for immunotherapy approach for MPN patients. And so it came up with this hypothesis that mutated CALR is an MPN-specific shared neoantigen that could elicit anti-tumor immune responses. And can we go after that? Next slide, please. And so we saw that as a therapeutic opportunity to target this particular protein. And this is how the idea for this phase 1 trial came up, where we're going to use peptide-based vaccines in patients with myeloproliferative neoplasm who have CALR mutation. And of course, we have to start with phase 1 trial, where we're really doing a small trial trying to look at the safety and a hint of efficacy of this approach in patients. Next slide, please. And the rationale for this is, like I mentioned, is that the mutated CALR neoantigen is only expressed by the malignant cell, and therefore it should result in effector T cells, so part of the immune system that goes after infections or after malignancies. They should recognize it as something that shouldn't be there, and therefore they should go after these cells, and the hematopoietic stem cells that are carrying this mutation could be potentially killed by these T cells. And then so the hypothesis is that the mutated CALR vaccine will improve our CALR-specific T cell immunity in MPN patients that carry this mutation. Next slide, please. New antigen vaccines is not a new concept. So we didn't just come up with this for this particular trial. So there are many different trials in oncology looking at this approach because there are many different mutations that result in these new antigens that could be targeted by our immune cells. And so these vaccines are being studied in a number of malignancies, including colorectal, pancreatic, brain tumors, melanomas, and others. Next slide, please. So I'm going to show you some of the preliminary work that was done by my colleagues in the lab. Specifically, this work was done by Chen Su, Simon Boscas. And she basically, we have tissue bank from patients who donate their blood for lab research for all kinds of work. And here we took cells from patients with MPNs, and they were stimulated with peptides. So these are parts of the protein of the mutated CALR. And the point of the experiment was, can we help these cells respond to the stimulation with CALR, with mutated CALR protein? And what is shown here is that, yes, we can. We can get an immune response, which is here is interferon gamma production is an indication of the immune response. And yes, when we use the mutant peptides, we can get some responses. And next slide. What was interesting here is then she took samples from different patients that have the CALR mutation and looked, some of the patients are responding and some are not. Can we see a pattern? And it seems that the pattern is that the patient who had ET were responding the best, a little bit less for patients who had ET-related MF, and even less so for patients who had primary myelofibrosis. And our hypothesis here is that the reason this is happening is that sort of the more aggressive, the longer the patients have the disease, probably the more difficult it is to elicit this immune response. So we should really try to go for earlier disease if we can. Next slide. So we always want to have specific aims for our project. So here, the aim was to conduct a clinical trial of mutated CALR vaccination and to assess the safety tolerability of administering this vaccine to patients with MPN. And then, of course, we wanted to see if what is the efficacy, or at least some efficacy of this particular approach. So we're going to evaluate the clinical responses. And importantly, we're also going to evaluate immune responses. So we're going to collect samples from patients as they're undergoing this treatment. And again, our immunology colleagues in the labs will look and study what happens with the immune response and what can we learn from this and how can we adjust things to make it better and stronger and deeper response. Next slide, please. So just very briefly, the inclusion criteria for the trial is that you have to have CALR mutation, because obviously that's the key point here. And then we want to take patients who have myelofibrosis that are low-risk disease, so not advanced myelofibrosis. And this is because of the data that I've shown you before, that we think that they're more likely to respond earlier in the disease. We also taken patients with high-risk ET. The reason why it's only high risk is that these are the patients that typically require treatment per current recommendations. This is phase 1 trial where we're assessing the safety. So we don't want to take patients that have very early disease. And then patients have to have adequate organ function, such as liver and kidney, and have to have reasonable blood count. So platelet counts more than 50,000. This is more obviously for myelofibrosis patients, and so on. Next slide, please. The important exclusion criteria is that we don't want patients on other therapies that could affect the immune response. So ruxolitinib and other JAK inhibitors, as well as interferon. But we could have patients continue on their current therapy with hydroxyurea or anagrelide, because that should not affect the immune response. And this is basically the therapy. These patients would get the vaccine, which is injected. The first 4 doses that's given every 2 weeks, and then it's every month. And then after 6 months of treatment, we do an assessment where a bone marrow biopsy is repeated. I know how much everybody loves bone marrow biopsies, but it really gives us the opportunity to assess what's actually happening in the bone marrow, and assess the immune responses in these patients as well. And then if patients are doing well, they may go on maintenance where they get the treatment every 3 months. Next slide, please. So I will not bore you with the details of how clinical trials are done in terms of all the paperwork, and all the time that it takes to get going and then to conduct the clinical trial. But we've treated 3 patients initially, and as the protocol was written, because we're worried about safety, we had to do it slowly, one by one. We had a safety meeting where we assessed everybody's responses. There were no safety signals identified. We decided we can proceed with enrollment of more patients, which is what we're doing right now. So additional patients are being enrolled. Study drug was well tolerated. There were no dose-limiting toxicities. Most patients did well, and the adverse events that we see were mostly injection site reactions, fatigue, and bone aches. And we call them grade 1-2, which means that they either did not require any interventions, or at most they required over-the-counter medications for interventions such as Tylenol, for example, for some of the body aches. That usually happen right after the vaccine injections. No patients came off treatment due to adverse events. And now correlative studies are in progress and immune responses are being evaluated. Next slide, please. So in summary, there are limited data right now. There's limited immune-based therapy options in MPNs. We really would like to change that. And not just in MPNs and myeloid malignancies. So in these types of malignancies, we really don't have effective immunotherapy, as opposed to, as I'm sure the CAR T-cell therapies and checkpoint blockade, all these drugs that are being used for both solid tumors and lymphomas and things like that. So this is something that is being studied with currently with monoclonal antibody approaches, this vaccine approach, and another vaccine approaches that are being done. And I think what's really important is that the study will allow us to expand our knowledge about what is the immune system doing in MPN. How is it involved in disease progression? Is there any way that we can harness it better to really control this disease earlier in the process, before it really becomes so aggressive that the immune system really doesn't have a chance of controlling it. And of course, this work is possible due to generous support from MPNRF. Next slide, I think one more. And of course, I would like to thank MPN Research Foundation for their support, but also many, many people who have worked on this project before me and with me, specifically Dr. Hoffman, Dr. Bardwaj. Like I mentioned, Dr. Simon Boscos did and will be doing all the laboratory work, as well as Dr. Camilla Yanko-Rubin, who was the MPN researcher who started this project, and many of my colleagues here who are working on this. Thank you so much for your attention.
Thank you so much, Dr. Krimenskaya. We very much appreciate your insight and all of your time today. So all throughout this webinar, everything that we do at MPN Research Foundation is for one purpose, and that's supporting patients, whether it's funding research, creating resources. It's all about improving outcomes in the lives of those living with MPNs. So I'd like to once again highlight that today on this Giving Tuesday, we have a chance to make an even bigger impact, because thanks to our $75,000 matching gift, every dollar you give will be doubled. So that means your support means more than just funding. It means better access to care, more educational tools, a stronger support network for patients and their families. When you give, you're not just donating to research, you're investing in real, tangible support for people facing the challenges of MPNs every day. And, together, we really can ensure that patients have what they need to navigate their journey with strength, confidence, and hope. And because we are focused on improving the outcomes of patients, I wanted to take a moment to encourage you, if you are a patient, loved one, or a caregiver, to take advantage of some of the resources that are available to you, though many of you may already be familiar with these. One of these resources is the National Comprehensive Cancer Network Guidelines, the NCCN. This document was recently updated and released in June of 2024. We can share the link for the electronic copy, or printed copies can be ordered through NCCN directly. But this guide, it's very comprehensive with information presented in plain language with visuals, charts, and definitions to empower people with MPNs and caregivers to talk with their clinicians about the best treatment options available. And of course, these guidelines are also available in many different languages. I also want to highlight one of our partners, which is the MPN Advocates Network. The Pathways program is in collaboration with this group to ensure we are educating patient and caregivers globally. The mission of the MPN Advocates Network is to enhance the ability of global patient advocacy organizations to improve patient care, quality of life, patient empowerment, for all patients who have been diagnosed with MPNs and their caregivers. And this group has close to 50 members representing their countries across the globe. Now, I did just mention the MPN Pathways program, and I wanted to make you aware of another webinar that is going to be taking place next month. This is actually going to be session 3 of our MPN Pathways series. The topic for this webinar is MPN Clinical Trials 101. It's going to be taking place on January 23rd, 2025, the same time that this webinar is taking place, from 5 to 6 p.m. Central Time. And during the session, participants will be able to review a basic overviews of MPNs and the role of clinical trials in advancing treatment options, learn the phases of clinical trials, the process of conducting them, and the criteria for patient participation, including potential benefits and risk. Also be able to learn the significance of clinical trials in developing new therapies and improving outcomes for MPN patients. Be able to learn about reliable information sources for those considering or involved in clinical trials, and then also be able to learn methods to engage with ongoing research, advocate for clinical trials, and support MPN patient communities. And our expert speaker, is going to be Christina Prasad, who brings 17 years of clinical research experience to her role, fueling her passion for advancing MPN research and clinical trial awareness. She leads the efforts here at MPN Research Foundation to engage healthcare providers, patients, and community oncology sites, fostering collaboration to drive meaningful progress in the field. And if you do have any questions that are related to patient or caregiver inquiries, please feel free to email patientengagement@mpnrf.org. Now, as Dr. Kremenskaya mentioned, there is so much happening in the research field focused on MPNs, and we're incredibly excited to be a part of this progress. So this is really the perfect time to talk about how we are looking to drive progress in 2025 and beyond. And as we look ahead, the future of MPN Research Foundation is really rooted in 3 powerful pillars: investing in research, convening the community, and advocating for patient voices in research. First, let's talk about investing in research. Our commitment to transformative initiatives like the MPN Progression Registry and MPN Challenge continues to pave the way for a deeper understanding and more innovative treatments. We'll hear more about the Progression Registry shortly, but by focusing on emerging research topics and special projects, we're not just keeping up, we're actively staying ahead. So from the clinical trial protocol reviews to raising awareness and recruiting for these trials, we're ensuring that research remains dynamic and patient-centered. But of course, research can't thrive in isolation, which brings us to convening within the community. We're creating spaces where collaboration leads to progress. So the MPN Roundtable in April of 2025 and the Progression Research Network Summit later in the year will bring together leading minds, scientists, clinicians, patient advocates to exchange ideas and push forward together. These gatherings are really more than just events. They're catalysts for innovation and change. And then, of course, we remain dedicated to advocating for the patient voices in research. Through programs like MPN Pathways and our UN MPN awareness campaigns, we're amplifying the experiences of patients and caregivers. So by integrating their perspectives into our research strategy and engaging them through communications and fundraising campaigns, we're not just advocating for better science, we're advocating for a better future for everyone impacted by MPNs. And these efforts position us for a future where the patient experience is central, research is bold, and the community is stronger than ever. And as we consider the future of MPN research and care, it's important to really reflect on the scope of the challenges that we face. According to SEER, the program of the National Cancer Institute, approximately 20,000 people in the US are diagnosed with an MPN each year, and around 300,000 people are currently living with an MPN. However, we believe that those numbers are much higher today, and that's just part of the picture. We also know that many people are living with an MPN but remain undiagnosed. So this gap in understanding highlights a critical need: better data, earlier detection, and more precise insights into how MPNs progress over time. And that's where the MPN progression registry comes in. By collecting real-world data from patients, we're building a more complete and accurate picture of MPNs, how they develop, how they evolve, and how they can intervene earlier and more effectively. This isn't just about numbers, it's about understanding the patient journey and transforming care for those who are currently diagnosed and those who will be in the future. So at this time, I'd like to invite our CEO, Kapila Vaijas, to speak more about the progression registry. Kapila?
Art, thank you so much. And thank you, Dr. Kremenskaya, for that amazing presentation about where we stand with the current programs, because as Clark said, it's a very exciting time for us in MPNs. While there are more options than ever, more JAK inhibitor options than ever for patients that are living with an MPN, we're also hearing about these promising new possibilities for targeting the CALR mutation. And as exciting as it is, and as grateful as we are for all the work that's been done by this community, supported by many of our generous donors, there is still so much more to learn and understand, and specifically about disease progression, as both Dr. Kremenskaya and Clark have alluded to. And so as a research foundation, we have really been focused on studying MPN disease progression, specifically trying to get at how can we better understand which patients might progress, what are some of the risk factors we would be looking at, and how would we know? And when we think about progression too, we're also thinking about, there are some clear definitions of going from, say, ET to MF, or from MF even to maybe, unfortunately, to acute myeloid leukemia. But there's a lot more along that journey, along that path of these chronic cancers that would lead us to better understand, is it possible for us to intervene earlier, to slow or stop or even reverse some of that progression, as Dr. Kremenskaya had alluded to. And so, as a foundation, we've been very focused on trying to help uncover this. And as we've explored it, this topic of progression continues to be top of mind across the board as a critical unmet need. And as Clark showed you, we sort of sit at this intersection of patients, research, and industry. And as we continue to hear from them, it is a very, very consistent, loud cry from every stakeholder that patients and their families continue to worry about that uncertainty, the unpredictability of knowing if they will get worse, and when, and how will they know. Because MPNs are, first of all, chronic. Patients can go for decades in relatively stable health, and then quickly, unfortunately, decline because of the heterogeneity that Dr. Kremenskaya mentioned as well, meaning the differences in the biology and the differences that drive how patients experience their symptoms, the differences in how they feel and their blood work changes. It's a lot harder across MPNs to find those patterns across patient populations, to better understand which patients might get worse and progress, and which will continue in that relatively stable health for some time. And so we really have to start to understand how to segment that better, but it continues to be a concern for patients. And then we all hear, as I'm sure many of our in our community who are patients and their loved ones experience yourself, that information can be scarce. There are gaps across the provider networks and the ways in which doctors treat and share information and understand MPNs, and specifically how they understand and talk about disease progression can vary from patient to patient and center to center. And so all of that creates that worry and uncertainty for patients. We hear that loud and clear, that we are being called to action to do something about it. And then as we talk to our researchers and clinicians, these physicians, they're also looking for more definition, more consensus about about how we understand progression beyond the very clear, progression to AML. What are those signals or biomarkers along the way before those transformations happen that they should be looking for to try to understand and help their patients through their own risk of progression. And so the researchers in the clinical community have told us that we just need more data. We need larger scale studies. We need to be following patients for a lot longer. We need to understand the natural history across larger swaths of patients. And right now, we just don't have that level of data consistently and continually to be able to come to that consensus and define that more completely. And then as we think to the industry, those folks who are actually bringing the later stage treatments through the regulatory process, through the late stage clinical trials, and making them available in the clinic to patients, they're looking for that better understanding and ways in which that they can use those, develop their programs for targeting new clinical endpoints that are recognized by the regulatory authorities and understanding the real world impact of the treatments that are out there. And so consistently across the board, all of the stakeholders have said, MPN Research Foundation, you are perfectly positioned to do something about this. And ultimately, we need more data. We need it more consistently for longer periods across larger numbers of patients. And so the next slide, Clark. After working with the community for several years to try to define it, it has become clear that we need to come to the table with a solution that works for everyone. And that means that we are going to be launching the first of its kind real world evidence MPN patient registry, which will be consisting of patients across the essential thrombocythemia, polycythemia vera, and myelofibrosis communities and indications, anchored by the study of disease progression. And when we say real world evidence, what we really mean by that is data that is collected and documented outside of the rigor of a defined clinical trial. Many folks have participated in clinical trials, and we know that they are very methodical and contained and defined. But what we're looking at is, how do we capture more robust data, more larger swaths of data of what's actually happening in the real world as patients are getting diagnosed, as they're getting their treatments, which treatments are they on, why are they potentially starting and stopping treatments, what are the symptoms that align with that, and then tracking that over time to see how that might potentially correlate with their journey and potentially progressing of the disease or not. And that would hopefully give us some clues. And so we are embarking on this journey over the next year to launch a database where we will be directly collecting electronic health records from patients, to over thousands of patients, seeking permission, seeking consent to bring those into a database directly from the providers where those health records are held, to start to give us that picture, to start to build that clue, that give us the clues as to where where we can look for ways to intervene with progression. And so this database is being built, as I said, over the next year. In the first half of 2025, We will be standing up the infrastructure. You can imagine trying to collect this type of data for thousands of patients over several years is not a small task. It does require some infrastructure, some investment, and some really thoughtful and careful planning, because what we want is to make sure that the data that we do collect is not only usable and recognizable by the research community, but also by the regulatory agencies to be able to use towards their decision making and making decisions about new treatments. And so the infrastructure is being designed as we speak. We are hoping to stand this up towards the middle part of 2025, at which time we'll be coming directly to the patient community and educating and engaging and providing access to resources as to how to participate and provide us the consent so that we can grab the feeds directly from your providers of electronic medical records. And then once that infrastructure is built, our hope is we can start to add layers of data on top of that. So for example, once we have an electronic medical record, we could potentially come out with surveys for patient-reported outcomes and ask the community about symptoms. Are you experiencing headaches and fatigue and other things that might correlate with the journey of the disease that we can pull into the medical record and see, well, how does that correlate with, for example, blood work and labs over a certain period of time? Ultimately, for us to study MPN disease progression, we are going to want to be capturing data like gene sequencing. A lot of folks get their gene mutation tested early on at diagnosis, but not always over time at different intervals. So we want to be able to influence and collect that type of data as well to see how do the genetic mutations change over time across that patient's journey. Or bone marrow fibrosis. We heard Dr. Kremenskaya talk of that as well, as how does the bone marrow change over time with treatment or without treatment? And what does that tell us about disease progression? That type of data is not necessarily easily accessible or ready to find, but once we've built this infrastructure and engaged the patient community, engaged the provider sites to pull some of the basics in, our hope is that we'll be able to add those insights and ultimately look for those clues, those markers, those signals of really understanding which patients are potentially going to progress and start to intervene earlier, and those that can live relatively stable and healthy lives and be more prescriptive about how they're choosing their treatment journey. So that's ultimately our goal, is to build out this registry. It's pretty aspirational for us as a foundation, but sitting at that intersection of patients, research, and industry, we feel pretty strongly that we're perfectly positioned to do it. We have the community support. We have the support of the biopharmaceutical industry. We know that the patients are asking for this. The caregivers are asking for this. It is the next sort of frontier in data science to break through for the life sciences. And so we're honored to be called to action to do it and take on this large task. And we hope to be talking more about this in 2025 as we go into our 25th year, reach out to the rest of the community and ask for your engagement and your support to drive more answers. I know everybody's looking for answers, and we want to facilitate that. So we're very excited for what's to come as we look ahead. Clark, next. I think there's one more slide. Is there a slide?
No, that was it.
That was it. Okay. So I'll end with thanking you all for your support to date. And, on this Giving Tuesday, we have the opportunity to acknowledge the support we've had so far and the excitement we have for what's yet to come, and looking forward to the community engagement as we forge ahead. So thank you. Thank you, Clark.
Thank you, Kapila. And yes, to echo that, we are eternally grateful to all of you for all of your support, for being part of the community, because we aren't able to do what we do without you. And your support, whether it's through donations, plan giving, volunteering, all makes a profound difference in advancing research, providing vital resources, and improving the lives of those affected by MPNs. So I want to encourage you to get involved as we wrap up, and there are a lot of different ways that you can do this. So you can donate through one-time gifts, recurring donations, or even matching gifts, which double your impact. Now, if you're thinking about a longer term commitment, you can invest in the future through plan giving, stocks, securities, or beneficiary designations. And then for those of you who are looking to take a more hands-on approach, we welcome you to volunteer, host peer-to-peer fundraising events, become an advocate, or connect with others within the MPN community itself. By joining this passionate community, you're not only supporting a cause, you're driving real progress and helping to create a brighter future for MPN patients around the world. Every contribution, no matter the form, brings us closer to that future. So, as one final reminder and call to action, I did want to reiterate that all gifts received today on this Giving Tuesday will be matched up to $75,000, effectively doubling your impact. That is all that I have for you today, but if you do have any questions, please feel free to reach out to me directly. You can reach me at giving@mpnrf.org. And once again, if you have any patient or caregiver inquiries, please reach out to patient engagement at mpnrf.org. Excuse me. Thank you all. Have a good night.